All right, well, hello everyone. Very nice to see you again. It's my pleasure to welcome you to the update on immunotherapy session, which will be presented by Dr. Loretta Nastapil and Dr. Jeremy Abramson. Dr. Nastapil is Associate Professor of the Department of Lymphoma at MD Anderson Cancer Center, a member of LRF's Scientific Advisory Board, and a steering committee member of our 2024 Follicular Lymphoma Workshop. Dr. Abramson is Director of the Lymphoma Program at Massachusetts General Hospital Cancer Center and Associate Professor of Medicine at Harvard Medical School. He's also the Chair of the New England Lymphoma Rounds Steering Committee and Co-Chair of the LSRMP 2024 and 2025 workshops. So thank you both very much for being here, and I believe we're starting with Dr. Jeremy Abramson.
Thank you very much. Good morning. Happy Sunday. What a treat to be here. Thanks to the LRF for convening us all together, for inviting me to share the stage with my friend and esteemed colleague, Dr. Nastupil. And so it'll be our pleasure today to talk about some advances in how immunotherapies have really proved transformative events across the range of lymphomas. And we'll take you through some of that today in what we can accomplish in an hour. And what we'll focus on are the most sort of more potent, most advanced, most exciting classes of immunotherapy today, which are CAR T cells and bispecific antibodies. So as a general level setting, what is immunotherapy in terms of lymphoma? Immunotherapy, quite simply, is the ability to use drugs or other medical techniques to harness the power of the patient's own immune system. Historically, whether it be chemotherapy, drugs, or small molecule inhibitors, most cancer treatment has been directed at the tumor cell itself. Immunotherapy's goal is to enable the body's own healthy immune cells to do the work, instead harnessing the power within to destroy lymphoma cells. Now, we've been doing immunotherapy for lymphomas for actually a very long time. But the challenge with immunotherapy is to create a very specific immune response against the lymphoma and not an immune response that can be misdirected against the body's healthy cells. So probably the first immunotherapy we've been doing in lymphoma for decades now has been allogeneic stem cell transplant. This is in patients with very advanced lymphomas historically, where we would use a donor's immune system, insert that into the patient after some low-dose chemotherapy, and try and count on the fact that that the donor immune cells will recognize the patient's lymphoma cells as sufficiently foreign and thus eradicate them. But it's also not a specific immune response. Those donor stem cells can also recognize the patient's bowel, the patient's liver, the patient's skin, the patient's retina, as foreign tissues and destroy that. So it is not a specific immunotherapy approach. And it's therefore one that requires long-term immunosuppression and carries risks of toxicities related to immune attack against the body's healthy structures. The real major advance for lymphoma therapy came at just around the turn of the millennium, with the FDA approval in 1997 of rituximab, the first anti-CD20 monoclonal antibody, really ushering in the era of immunotherapy and proved, I would say, until recently, the most potent and dramatic advance in how we approach lymphoma in the modern era. Rituximab and subsequent generations of monoclonal antibodies are part and parcel of both frontline and generally subsequent lines of therapy across the B-cell lymphoma spectrum. Now, rituximab, obinutuzumab, tafasitamab, these are what we call passive antibodies. These are antibodies just like our own bodies produce. Our bodies produce antibodies against bacteria, viruses, pathogens of all kinds. And so some smart people in a laboratory several decades ago said, why don't we create an antibody against a lymphoma cell? They did. They targeted a lymphoma against the antibody against the CD20 antigen, which is on the surface of lymphoma cells, and they give that intravenously, and now we can do that subcutaneously. The way antibodies work is by binding to the lymphoma cell and then counting on the body's own healthy natural killer cells to stumble by, recognize that, and act against the lymphoma cell. It's what we would call a passive immunotherapy. It counts on the body's immune system really recognizing and playing along. And of course, resistance can develop to this type of treatment just as they can to many other types of treatment. But this has been a benchmark for what we've been doing for a very long time. So here's an example of a lymphoma cell. There's a cute little molecule of rituximab floating on in. And what we count is that a natural killer cell then recognizes that and then induces cell-mediated cytotoxicity with the body's own NK cell. That's a healthy immune cell, and thus destroying the cell. And that proved transformative. Now rituximab combined with bendamustine for follicular lymphoma, CHOP for more aggressive lymphomas, EPOCH for even more aggressive lymphomas is now standard of care. And then we started to think, can we make immunotherapies work even better? Now, one way is we can use these antibodies as effectively drug or radiation delivery systems, use these as ways to target those drugs towards the lymphoma cell. This leads to things called antibody-drug conjugates, and you'll know these potentially as drugs like polatuzumab vedotin, brentuximab vedotin, more recently a drug called loncastuximab tesirine, as well as a rarely used drug that delivers radiation therapy to cells called ibertumumab tioxetan. And then there's other ways we can try and activate the immune system by helping unmask tumor cells from the body's own immune cells, something called immune checkpoint inhibitors, something that's extraordinarily active in Hodgkin lymphoma, uniquely among lymphoma subtypes. But we obviously still need to do better today, even with all these existing tools that are currently part of our armamentarium. Diffuse large B-cell lymphoma and other high-grade B-cell lymphomas, the most common lymphomas in the United States and worldwide. We cure the majority of patients with these diseases up front, but about 30% of these patients will still relapse after initial therapy and are in need for curative therapies thereafter. We've had dramatic improvements in the treatment of mantle cell lymphoma with optimization of chemotherapy, novel targeted therapies, but the majority of mantle cell lymphomas today are still considered incurable and life-threatening in a significant proportion of patients. We think of follicular and other indolent lymphomas as very favorable prognostic diseases, and they are. About 80% of patients can expect a normal life expectancy with these diseases, but that means a subset of patients are not assured a normal life expectancy and are at risk of even dying of those indolent diseases where we need to do better. And the same is true despite dramatic advances in targeted therapy for CLL. So this takes us to the next generation of immunotherapies, which are the purpose of our session here today. We can do CAR T cells. CAR stands for chimeric antigen receptor. This is where we genetically engineer a patient's own T lymphocyte to express a receptor against their lymphoma. We create that in a laboratory. We administer that back to the patient and let the patient's own T lymphocyte, now bionic, engineered and structured to target their lymphoma, go in and do the work. Unlike prior cell therapies like allogeneic transplant, an engineered T cell is a patient's own T cell, so it doesn't have the risk of targeting their other healthy tissues, and it's a one-time treatment. It's a living drug that can then grow, divide, and persist in circulation and does not require immunosuppression long-term like transplant does. Bispecific antibodies, I'll show you, and Dr. Nastupil is going to show you in great detail how these antibodies are ways to create an off-the-shelf mechanism for harnessing the power of a patient's T cell. So how might we build on what I previously showed you as a passive antibody? In this case, you've got a lymphoma cell, but let's create, instead of a single-headed antibody, a two-headed antibody. Now you can see that antibody is going to directly bind the patient's T cell in a targeted way. Effectively, this antibody binds to the tumor cell, throws a lasso around the patient's own T cell, brings it in for a kill me kiss, and you can see the T cell eradicates that lymphoma with an off-the-shelf drug. What about a chimeric antigen receptor modified T cell? This requires a bit more work. We have to create this receptor in the laboratory. We then have to insert it into the patient's own T lymphocyte, here with a bolt of lightning, but in the laboratory it's actually with an inactivated virus. That then leads to this new receptor now being expressed on the surface of the patient's own T lymphocyte alongside their normal T cell receptors. So a patient's own T lymphocyte now has receptors against the tumor cell as well as their normal T lymphocytes that now can directly target the lymphoma cell and destroy it. So two highly potent, very distinctive methods of activating the T cell. The CAR T cell process does require a bit of to-do to create this. We need to collect a patient's T lymphocytes and then genetically modify them in the laboratory. Once we do that, they're grown in the laboratory to create a fixed amount that we can administer back to the patient. Once we have these CAR T cells generated, we prove that they're safe, that they're effective, that there's a good number of them, that they work. We then have to give some low-dose chemotherapy because these cells are the patient's own T cells, but they're slightly modified because they have that chimeric antigen receptor on their surface. And that alone can generate an immune response. So we have to give a patient some low-dose chemotherapy to prevent these CAR T cells from being rejected. We then administer back to the patient, and those cells can then go in and get to work. Now, this was sort of a science fiction story until now well over a decade ago, when a young girl named Emily Whitehead, who's been very public in her story at the University of Pennsylvania, had multiply relapsed pediatric acute B-cell lymphoblastic lymphoma, or ALL. She had very advanced disease. She had no standard treatments left, and she became one of the first patients enrolled on a pediatric CAR T-cell trial at the University of Pennsylvania. Her disease went into a remission on her Facebook page every year. She's posted the number of years that she's been cancer-free. At 10 years now, she highlighted her disease by saying cured. I think strikingly ushering in the power of this potent immunotherapy in a young woman who clearly would have otherwise succumbed to her disease and is now an ambassador for this highly potent, highly successful, transformative treatment. We all have our own stories of the first patients that we treated with CAR T cells. The first patient I treated was before I had access to a CAR T cell. This was a patient who I met in 2011, 42-year-old young woman, mother of three beautiful young children. The oldest was 8 at the time. She had primary refractory primary mediastinal large B-cell lymphoma. Her disease progressed on dose-adjusted EPOCH-R and on 7 subsequent lines of therapy, including 3 clinical trials. At the time, I said, there's this really interesting trial happening at the National Cancer Institute exploring a type of therapy called a CAR T cell, which would subsequently be licensed to Kite Pharma and become axicabtagene ciloleucel. I referred her to the NCI, where she became, I think, patient number 6 on that clinical trial. Her disease was highly refractory in the mediastinum. You can see a big tumor in her liver right there, as well as involving her kidneys. This is all disease. And 30 days later, she was in a complete remission. That was 2013. 5 years after that, she gave a toast at my wedding. And a few weeks ago, I saw her socially because she's long since cured of her disease. And for me, said, this is no humbug, and we all need to start investigating this treatment and bring this closer to patients. And this is incredibly transformative treatment. It certainly has been for her. And that single patient is what got me excited about CAR T-cell therapy, which has now been my dominant research focus for the last decade, really since that time. So it's transformed not only patient lives, but my own humble little career as well. So where do we use these treatments today? Well, right now we're investigating them across a large panoply of lymphomas, but they are truly transformative, fully FDA-approved therapies in relapsed large B-cell lymphomas, follicular lymphoma, and mantle cell lymphoma. I'll highlight some of these areas for you today without going into too much of the weeds. Relapsed refractory diffuse large B-cell lymphoma, as I mentioned earlier, is the most common lymphoma. About a third of patients will relapse. Historically, the standard treatment for relapsed diffuse large B-cell lymphoma was giving more chemotherapy. If that chemotherapy worked, we gave even more chemotherapy after that, called high-dose chemotherapy, and on autologous stem cell transplant. So to give you a sense of the state of the field, for decades, those of us in this field would say, oh, wait, chemotherapy isn't working. I've got an idea. Let's give you more chemotherapy. Let's throw good money after bad. And you know what? It didn't really work very well. It turns out fewer than a quarter of patients would be successfully cured with that approach. It's the best we had at the time, but obviously progress was needed. And where it worked the least were in patients who had the most chemotherapy-resistant disease. 3/4 of patients who relapse after initial treatment today with diffuse large B-cell lymphoma will either not respond at all to chemotherapy or relapse within or progress within 1 year. The likelihood is close to 15% for a successful outcome with just more chemotherapy. It's into this very context that we've started evaluating CAR T-cell therapy. And so the first FDA approval for CAR T-cells were in patients whose disease failed to respond to initial therapy, then relapsed again after that, and in patients with multiply relapsed diffuse large B-cell lymphoma with a median of 3 or more prior lines of therapy, meaning patients who were getting their 4th type of treatment for large B-cell lymphoma. No fewer than 3 CAR T-cells have been FDA approved in that context. Those are axicabtagene ciloleucel, or axi-cel, lisocabtagene maraleucel, or liso-cel, tisagenlecleucel, or tisa-cel. How in the world they come up with these absurd names and make us all learn how to say them? It's criminal. But the drugs work, and so we're stuck using these names. And no fewer than 3 of these CAR T-cell products are now fully FDA approved for patients with 3rd-line or later large B-cell lymphomas. Today, we primarily use axi-cel and liso-cel. These have the highest response rates. In this context, historic therapies would have had a complete response rate in the range of 5 to 7%. These drugs have complete response rates in more than half of patients. And if you look more than 2 years later, just more than 40% of patients remain progression-free with their disease previously unheard of with conventional therapies. It's also a reminder if 42% of patients are progression-free at 2 years, that means nearly 60% are not. And so we obviously need to make this treatment work better for more patients than they are now. It's a dramatic advance, but I think we are still at our infancy for how we employ these treatments in the care of our patients. We now have 5-year follow-up on the ZUMA-1 study, which was the study that got the first FDA approval for axi-cel. And what you can see in this Kaplan-Meier curve effectively is flat after the first 6 to 12 months, which means that there appear to be no further relapses, even out 5 years, once patients are in remission at the 1-year mark, showing that this is not only a treatment potent enough to induce complete responses, but those responses are indeed long-term, and thus we would consider curative therapy with fewer than half of these patients actually dying of their disease even 5 years later, previously unthinkable. Now, this therapy doesn't come without side effects. All treatments do. The side effects of giving a highly potent T-cell therapy is that we activate the immune system. We literally give T-cells, and then we turn them on and harness their power against the lymphoma. And that means you get this massive immune response, this immune stimulation. Think about sudden inflammation in the body. And what are the hallmarks of inflammation? Fevers. It can lower blood counts. It can create, so you might have heard of people who are very high fevers and they get kind of delirious or confused. We can see that with CAR T-cell therapy. We deplete out healthy B lymphocytes in the process, and so we can see low B-cell levels and low antibody levels. These are all manageable, all reversible, all recognizable. None of these we consider obstacles in the treatment of patients, but they require attention, which is distinct from other non-immunotherapy approaches. So these are the toxicity levels. There are differences in the products, and I'll tell you that axi-cel is the more toxic of the other products. There's biologic reasons for that. Axi-cel has higher rates of cytokine release syndrome, meaning this inflammatory state, higher rate of neurologic toxicities. These are now usually lower grade, meaning mild. They're usually reversible, and so these can be factors in considering the selection of treatments among patients. But importantly, despite the differences in the toxicity profiles, they're all active and the toxicities are manageable regardless of the underlying product. So while CAR T-cells first came into our treatment armamentarium in patients who'd had multiple prior therapies for large B-cell lymphomas, what if we cut out that earlier phase of chemotherapy and use this after the very first relapse of large B-cell lymphoma? And so if historically we used high-dose chemotherapy, as I mentioned, that successfully treated fewer than a quarter of patients, and that many of patients might not be even considered candidates for a high-dose chemotherapy approach, what if instead of doing that historic standard, we do CAR T cells instead? And now no fewer than 3 randomized controlled trials have said, let's take patients who with first relapse of large B-cell lymphoma after their initial R-CHOP or their dose-adjusted EPOCH-R, and they now have primary refractory or relapse disease within 1 year of that initial treatment, which is the majority of patients today, and we'll randomly assign half of these patients to get our prior standard, or at the time our current standard, meaning more chemotherapy, and then more chemotherapy after that, and a stem cell transplant, or give them CAR T-cell therapy and supercharge their T-cells. Well, now 2 of these trials have proven to substantially improve outcomes compared to the historic standard of care. The trial of tisagenlecleucel did not do that. And so based on these trials today, we know that both axi-cel and liso-cel dramatically improved complete response rate, event-free survival, progression-free survival, and overall survival when you adjust for risk factors. That shows us that we are inducing deeper remissions. Those remissions are lasting and indeed are durable and more curative than our historic standard of care. This is now currently FDA-approved therapy. Now I would say wherever you are in the country, the best treatment for relapsed diffuse large B-cell lymphoma after failure of frontline therapy would be CAR T-cell therapy. Part of the problem is these aren't available on at every hospital near where all patients live. Sometimes it requires traveling to get to this treatment, but it is substantially better than the historic standard of high-dose chemotherapy and transplant. And we also look at not only cure rates and complete response rates, but quality of life metrics. Quality of life is better with a CAR T-cell than just throwing chemotherapy after chemotherapy after chemotherapy, which is good to know. What about patients who aren't candidates for transplant in the first place? We do have a trial that said, not all patients are even candidates for high-dose chemotherapy. What if we've got older patients who we might not want to give high-dose chemotherapy to? They wouldn't have been eligible for those randomized trials I showed you. This study of lisocabtagene maraleucel as second-line therapy in non-transplant eligible patients was primarily due to being age of over 70 years old or having other medical problems, which is a significant proportion of patients with large cell lymphoma, showed that the complete response rate is very high as well, and that we're seeing a cure rate of just in the range of 40%. Again, the FDA therefore did approve liso-cel as a second-line treatment for non-transplant eligible patients, and that's independent of duration of initial response to, say, upfront R-CHOP treatment. Now that's in large B-cell lymphoma. What about follicular lymphoma and other indolent lymphomas? These are the second most common type of lymphomas in the United States and worldwide. And guess what? CAR T-cells work here as well. There are now no fewer than 3 large phase 2 trials which evaluated CAR T-cells in patients with multiply relapsed follicular lymphoma. These are patients with 3 or 4 prior lines of therapy for their low-grade lymphoma, where we have limited standard treatment options at the time. We now have both bispecific antibodies and CAR T-cells available in this space. But if you look at these patients where previously few options were available, axicabtagene ciloleucel inducing complete response rates in close to 80% of patients, with 2/3 of patients remaining progression-free even a year and a half later. Tisagenlecleucel or tisagenlecleucel, complete responses in over 2/3 of patients, with more than half of patients remaining in remission with no evidence of disease 2 years later. And most recently, the data for liso-cel was presented at a research meeting in Switzerland, and the study of liso-cel in over 100 patients showed a 94% complete response rate with limited follow-up at a year, over 80% of patients remaining in remission and progression-free. These are heavily pretreated, 3-4 prior lines of therapy. Most patients with follicular lymphoma will never get 3 or 4 prior lines of therapy. but some will have 7 or 8 and have steadily reducing success rates. This is truly game-changing therapy for patients with multiply pre-treated lymphomas with ongoing follow-up and ongoing studies. What about mantle cell lymphoma, another potentially high-risk lymphoma, particularly in patients who have upfront chemotherapy and then have a drug like ibrutinib, zanubrutinib, or acalabrutinib? Patients in the multiply relapse setting historically had fewer highly effective treatment options. Here, 2 CAR T cells, Axi-cel, which is approved, this should say brexucabtagene autoleucel, not axicabtagene ciloleucel, but Brexucel is now FDA approved for relapsed mantle cell lymphoma. The Liza-cel data recently presented as well, but not currently FDA approved, showing, again, high response rates with durable remissions now with 3 years follow-up, even in patients who've had 3, 4, or 5 prior lines of therapy. So really deep remissions, long-lasting remissions in patients just by harnessing the power of the immune system. So these are incredibly game-changing treatments across multiple B-cell lymphoma histologies. We do think beyond just celebrating the success. When we achieve success, we think about long-term consequences of depleting out B lymphocytes, of decreased antibody levels. Patients are at risk for ongoing infections, even potentially years later. So it's important that we pay attention to that. It's important to get vaccines, COVID, influenza, RSV as we get into enter the fall season. It's important to work up fevers with attention to potential even uncommon infections, and to give back antibodies as a replacement treatment where needed. We're trying to move these technologies further to reach more patients with other lymphoma histologies, marginal zone lymphoma, Waldenstrom's macroglobulinemia, chronic lymphocytic leukemia. There's investigation across the board in these. We're trying to think how we make them work better, combining CAR T cells with other available drugs to soup up the immune response. Build a better mousetrap, make better, healthier CAR T cells. Build CAR T cells that we can actually take off the shelf from a donor, as opposed to having to take them from a patient who's already had multiple types of chemotherapy, and trying to take this treatment, make it work even better in the service of our patients for years to come. This is just one exciting advance immunotherapy, but I'm particularly excited to hear about the other advance, speaking of off-the-shelf products. So it's my great pleasure now to introduce Dr. Nastupil to talk about bispecific antibodies. Thank you.